Good morning, good afternoon. My name is Frédéric Bordes-Picard, and I'm the Pharmaceutical Marketing Manager in Europe, Middle East, Africa for Capsugel. Today, with my colleague Michael Burke, we will be providing a webinar on spray-dried process and formulation consideration for inhalable dry powders. Interest in pulmonary delivery of drugs is increasing for both acute and systemic disease conditions. Particle engineering via spray drying is an enabling technology for a number of new actives for lung delivery. Process and formulation attributes are tunable for either small or large molecules, range of physical properties, or desired delivery platform. Robust spray drying approaches have been developed to ensure phase-appropriate formulation development with streamlined timelines and cost. Encapsulation of spray-dried formulation can further enhance speed to clinic through commercialization. During this webinar, we will discuss how target product properties and profile can be met by altering process condition and excipient selection for inhalable dry powders. Michael is a senior research chemist and inhalation group lead at Capsugel band sites. He has helped lead multiple dry inhalation projects from feasibility through clinical application. He has extensive knowledge in inhalable dry powder formulation development as well as spray dry process development. In addition, Michael is our analytical expert in inhalable dry powder characterization. He has been with Band Research and Capsugel since 2010. He graduated in biochemistry from the University of California, and he holds a BS in molecular biology from the University of California, San Diego. We will be having a 45-minute presentation followed by around 15 minutes Q&A session. So do not hesitate to post any questions or comments that you wish using the message center bar. Michael, the floor is yours.
Thank you, Frédéric, and thank you to everybody for joining today's webinar. I hope the presentation itself will be informative and give you perspective on the formulation and process approaches we have for our spray-dried powders. Again, today's topic is spray-dried formulation and process considerations for inhalable dry powders. Prior to going through today's presentation, first off, I just need to mention the slides are here but are not for distribution and really are for purely educational purposes. If you have any questions or with further detail, feel free to contact myself or Frédéric at any time. And as Frédéric said as well, toward the end of the presentation, hopefully we'll have enough time for a Q&A session. So today's webinar will go through a couple of different topics. The first one here we're going to review is the Capsugel spray-dried particle engineering capabilities for inhalable dry powders. This includes an overview of the technology we provide, corresponding analytical characterization, encapsulation, and then how that actually feeds into product concept design. After that, we'll dive into more of a discussion around inhalable dry powder formulation selection and how that's based on physiochemical properties and target product profiles. After defining formulations, we'll talk about particle engineering via spray drying. We'll have a quick overview of spray drying itself and then dive into particle concepts really based on the target product profile that we defined in formulation. And of course, having a formulation and having a process doesn't really necessarily mean anything until you have a finished product. In this case, we'll talk about filled capsules or additional elements to do that for secondary packaging. At the end of the presentation, we'll tie this together through two different case studies. The first case study will look at differentiating formulations based on properties, and the second will look at the scalability and the ability to preserve properties across an API load. Again, I will provide as much detail as possible throughout the presentation. Of course, this is an exhaustive topic itself, and So if there are any questions, please bring it up toward the end of the presentation. So I'd like to start by just giving you an idea of Capsugel itself. It is a global company. You can see through the map that there's sites throughout the world. And really what this allows us to do is have a lot of connections throughout your site, and it allows for additional communication. I'm actually located in Bend, Oregon, the west coast of the United States. This site is focused on our spray drying technology and anything from early feasibility through commercial manufacturing. Now, Capsugel as a whole offers multiple platforms and has a wide range of knowledge and experience. These capabilities include the 4 different buckets here you can see on the slide, which includes bioavailability enhancement, targeted release, capsule technologies and encapsulation, and specialized applications. Of course, what is relevant to this presentation is our expertise in spray drying particle engineering for inhalable dry powders. And those capabilities include API characterization, ability to pre-formulate, design formulations for spray drying, performing spray drying feasibility, and this can be either small molecules or biologics. We have the corresponding analytics to go hand in hand when following through the spray dry feasibility studies. Of course, performing long-term and short-term stability studies. We have a clinical campaign manufacturing. This also includes now GMP release of those products. We also have the ability for commercial manufacturing of spray dried products. And then along with that, we do have a wide range of capsule capabilities and encapsulations, which we'll touch more in the presentation. So on slide 6 now, we're going to use this model a couple of different times in the presentation just to kind of orient us all. So we use this really as a holistic approach to dry powder product development. Each activity, as you can see through here, is actually performed on-site and routinely done in parallel. So we like to use this approach because it's rational and robust, enables kind of reliable product development. And as I said, I'm going to reference this throughout the presentation. But I kind of want to give you an idea before we move into it. So, of course, what we do here is we start with an active product concept. We feed in what kind of active that we're going to look at, and then mix that with the necessary materials to make sure we ensure stability and feed into process conditions. As I said before, analytical characterization is critical, and this is done in parallel when after formulating and engineering. Ensuring stability and moving up to scale and optimization is critical, and then understanding what your finished product is going to look like. In each case, what you want to do is deliver a phase-appropriate material. So I know most of you already know what is spray drying, but I kind of want to touch on this and why we actually use this for engineering particles. So spray drying itself is an enabling technology, has high precedence in the literature, and we've also been using it now for multiple years. What the particle engineering technique does is improve certain qualities of your powder that you're delivering. So just to orient you real quickly, we have a spray solution that's going to be mixed in now. You have your excipients and your API, which will define what your drug loading company uniformly may look like and overall particle size depending on the solid loading. Will be fed through an atomizer. This atomizer now has a couple different attributes to itself. You're looking at what is the spray pattern or spray plume, what is the size of those droplets, and how that defines your final particle size. But as the atomization occurs, you have a drying nitrogen gas that's mixed in to actually set up the thermal conditions in the spray dryer. The spray dryer will define then how the material evaporates and is recollected, goes through a cyclone in this case, where you separate the gas stream from the dense particles that you've created. And then in the end, you have kind of a particle collection, which will be the final product you use to define your aerodynamic size and density. So as I stated, really, there's a lot of value added by using spray drying for inhalable dry powders. Of course, a lot of folks have looked at crystalline lactose blends, either coarse or fine lactose, mixing with the API, you're limited there by low active loadings, and then you have to worry about the cohesive forces between the lactose itself. Now, using the spray drying process, you're engineering particles to be how you would like to deliver. So, of course, they have a specific size. They have defined physical properties. You can vary drug concentrations, which can actually allow for dose matching if you don't quite know what your dose needs to be. You have homogeneous particles. Each particle is exactly the same as you go through and collect. You can add functional excipients and create aerosolization and absorption. You have increase in stability. You can have combo therapies. It can be amorphous or crystalline. And in this case, depending on how you deliver the crystal material, you don't typically worry about the aspect ratios of those given crystals. You can move on to poorly soluble compounds. And then also a lot of the steps are drug sparing. In the end, what we're looking to do is improve a product that now allows for efficacy, reduces variability and side effects while improving patient adherence for simplicity. So now at Capsugel as well, while we're doing these steps, we're looking for full analytical characterization. And this needs to be done in parallel with manufacturing to really understand the kind of product you're creating. So understanding what the spray solution stability is, when you have your API that's either dissolved or suspended with the different excipients, how long can you actually hold that material prior to processing? Do you have any chemical degradation? Of course, now that you're creating dry powders, you need to understand the solid state analysis. What kind of water content? What kind of residual solvents do we have? What is the overall morphology, and how does that impact particle performance? We want to ensure that we're not getting any type of agglomeration or aggregation of some sort through those particles that are being collected. What is our physical state of the material? What is the TG or TC, so the glass transition temperature, or the rate of crystallization that's occurring if you have an amorphous form? Of course, because we have dry powders as well, and as we all know in the inhaled world, we're using a lot of materials that are hygroscopic. We need to know what is the actual rate of absorption of that water. And finally, as well as characterizing what is the actual particle size as a geometric size. We use the Malvern here. Chemical analysis goes hand in hand to ensure potency and purity. And then your aerosol characterization, the two different steps of understanding what is your content uniformity, and then also through the NGI, understanding what is your MMAD, EF, GSD, and fine particle fraction. And again, at Capsugel as well, we have encapsulation capabilities, so creating the dry powders themselves and then turning it into a dosage form. Two different encapsulators we use here are an Excipients 600S. It's a gravimetric fill, 100% weight checks, really is used for more development or feasibility through phase 1 and 2. We do have a large-scale encapsulator now as well, a Hera-Hoflinger Magi CMS. As you can see there, the capsule speed is quite high. We're looking at almost 70,000 capsules per hour. It's a volumetric dosing system, 100% weight monitoring as well, and we also have this ability to translate this from anywhere from phase 1 and beyond. So just to summarize some of the capabilities here we have in-house prior to moving on to the formulation and process conditions. We do have a lot of expertise in the spray powder development, anything from formulation to process to characterization, to scale up to encapsulation and GMP qualities or capabilities. Of course, this translates to multiple client advantages and allows for robustness, a path forward for your API, ability to retain stability, and have rapid progression through each step. So I'm going to transition here the next couple slides, and we're really going to focus on formulation development. And really, in each case where you care about a formulation, it's something that's, for one, if you're early phase, really have the ability to do in vivo dosing, understand your dose, your PK/PD profiling. If you're later stage, you care about really stability and overall performance and robustness. In each case, the goal of formulation is to establish that stability, the dosage, and provide the aerosolization that's necessary to get into the lung. And just in general, formulation concepts are really translatable to each API and then are fine-tuned based on those properties. And so you can kind of see on slide 12 here is just that top left section of that model that we presented earlier. What we'll go through is what the active product concept may be, and then how to actually design what the formulation is going to be based on the active material. So to start off here is what kind of compound are we looking at? Is it a small molecule? Is it a biologic? Of course, spray drying can handle either one of these. And it's very critical to understand what are the molecule properties, so we understand the charge of side chains, what is the overall secondary structure for a biologic, what kind of hydrophilic or hydrophobic residues does it have, what is the overall solubility, do you have high aqueous solubility, do you have low, what kind of organic solubility do we have for the small molecules. That actually feeds into what kind of excipients you can add and how you're going to process the material. Do we want to have a crystalline or amorphous form? Now, this is really going to be based a lot of times on PK, but also the physical or chemical properties of the molecule itself. Can it sustain an amorphous form? It doesn't need to move into a high-structured crystalline. Are we looking for local or systemic delivery? Do we have that target in the lung, or is it going to go ahead and pass through in the bloodstream? Again, what part of the lung? So that's going to help us define what kind of particle size we want to hit when developing the product. A lot of cases, there's wide dose ranges. We can have something that's very low active loading, so we tailor the process or tailor the excipients to that. Or in some cases where the neat formulation is necessary because the molecule itself is not terribly potent. And again, additional qualities we want to consider, as said before, is the Graston temperature if we're having an amorphous form. The melting temperature or also the crystallization temperatures are critical as well to understand how the molecule may behave during the drying steps. What is our given pKa if we know that? What is our log P and our corresponding dose? So the basic guidance a lot of the times is around PK. So what kind of pharmacokinetics do we need for the molecule? Do we need something that we want to have a high transfer rate? Do we need to have something that's going to stay in the lung for a longer period of time? So that's sort of defined a lot of times by the log P, or the given melt temperature will tell us a little bit about how is that high-order crystalline structure, how stable is that? So there's a couple different things here that we have on slide 14. On the left is more a general graph of log P versus melting point, and again will help define the formulation. On the right here now is your kind of BCS classification in a way of inhaled products. So what is our actual permeability, and then what is our aqueous solubility? So things that are biotherapeutics, of course, have typically higher aqueous solubility but lower transition across epithelial. And small molecules can be across these ranges. So, knowing what kind of solubility we're going to have and what kind of performance it's going to have in the lung is really going to help define, for one, what the physical state of the molecule that we need to have in the spray dried particle, two, what kind of excipients are we going to add. So as I stated before, there's two different physical models really for engineered particles. They can be either crystalline or amorphous. Now, when I say crystalline, this is typically not, thought of as your typical jet milled and mixed with carrier blends. You'll either have two different types of crystalline products. You can have something that's top down or bottomed up. So we can have a product that will be jet milled down and then suspended and put into an excipient matrix. Bottom down or bottom up is more of a way of creating crystals while utilizing the spray drying process. So understanding really what you need as a product profile is critical to understanding how you're going to translate that into a formulation and a process. So that helps to define what kind of flow-enhancing surfaces do we need, what other excipients that we need to add, binders, do we need diluents, what is our particle morphology just going to look like based on kind of these physical makeups. And you can see on the slide here, the top left there is a representation of what a crystalline spray-dried product typically would look like. Bottom right is an amorphous particle with a surface coating. And as I said, typical profiles, those with a high melting point, will lend themselves to a crystalline API with usually a correspondingly low TG. With low melting points, really it's more applicable to moving toward an amorphous formulation development. So we've touched on, what kind of physical situation we want to have with the API, and again, define on those properties within the molecule, or what does your dose need to be. So we have a toolkit of formulation excipients that we can use to help either provide stability, bulking agents, modifications of the particle if we feel like we need to have better aerosolization, or be able to decrease water absorption if we have something that's hygroscopic. Now, in each case, the primary functions, as I said, are really are what we're considering while doing the formulation. So you really want to have those excipients included only if necessary and there to deliver desired properties. If it comes to the situation where, again, we do not need excipients, the molecule itself is relatively stable as either a crystalline or amorphous form upon spray drying, or we need a high dose of the material, we'll go ahead and not use excipients and try and develop the highest neat formulations possible. In each case, a lot of times you have buffer components that need to be included, salts are fine, sugars, things like that, polymers. And again, you're focusing on what is your actual rationale. And those key properties are important then based on the rationale. So having the bulk in H, you really want to have something that's not hygroscopic. You're dealing with dry powders, could be exposed to water contents. Stabilization, either it's engineering or solid state. Do we have something that we need to protect the particle as it dries? Do we get that coating as necessary? And then for the stabilization of the API in the solid state, we have an interaction with the molecules to ensure there's no mobility. And that's a function really of the overall dispersion TG or mixture TG. For the physical state as well, we care about what is the high TG to limit mobility. We want to ensure there's no particle agglomeration. There are certain excipients that can help reduce the sticking upon storage, and that's really a function of the surface of the particles. And as I said before, too, aerosolization is key, right, as we're going through and engineering materials. So we have something that's hydrophobic that resides on the surface. So we're going to add those excipients in to define that particle. And in some cases, you can add mucoadhesive agents that will modify the PK or PD profile. So now there's a great paper that was published by Jeffrey Weirs back in 2015 that put a kind of a list of excipients together, and these are really precedented excipients about the literature and ones that we'll use as well. As you can see, the options are somewhat limited in what you can do, and that's really just a function of what the lung can tolerate. In a lot of cases, too, we're looking at combinations of these excipients that are listed in this figure. You want to be able to provide different elements to the particles as we define. So these are all things that will be added into with the API as we spray dry. So ones that we like to focus on a lot are the surface force excipients in this table. So you have your phosphatidylcholines, or your phospholipids here in this case, or your hydrophobic amino acids, which help to define the particle surface. Leucine is one that we use readily. Now, in each case, again, as I said, these reside on the surface during the drying droplet and to decrease cohesion as you collect the particles. So it's things that will go in together with the API. The phosphatidylcholines form as colloids on those surfaces. Additionally, what we want to have a lot of times for bulking agents are our glass formers. Now, this includes, or this increases the overall TG of the material in each individual particle. You'll have hydrogen bonding interactions here a lot of the times with the API itself. And again, the idea here is to decrease molecular mobility and establishing that higher TG. A couple of excipients that are on this list that we use readily are triglycerol and sodium citrate, along with PBP. Again, there are a number of excipients that you can use in this this figure, and it's really going to be based on what does the API need in each situation. We want to ensure stability. We also want to give the particle performance that's necessary through the spray drying process. So one example I'm going to use here on slide 18 is now leucine. So leucine is a great excipient to have within your formulation. A lot of the times you are limited a little bit by solubility, but what it's doing here is to control the surface and decrease electrostatics through kind of van der Waals forces. So as we set up a particle, what we'll have is leucine in the evaporating droplet will move to the surface of the particle, and it forms more of a hydrophobic shell over the materials inside the particles. Now, you can titrate the amount of leucine into the formulation based on what you kind of need for dispersion and also for your active loading. So on the left-hand side, really, is looking at increase in leucine content. And they use PVP in this case as just a model bulking matrix. And you can see as you increase small amounts of leucine, it does change the particle surface and particle morphology. On the right side here, now we're increasing higher amounts of leucine from just a given particle. We use trehalose, 100% trehalose on the top right there, and then increase 25 or 50% leucine. You can see those differences in the particle surfaces and the rugosity that's changing. And really what that does is help better disperse the material. And again, as I said, leucine is a hydrophobic amino acid. It can decrease water absorption of those individual particles. And of course, in each case, though, we'd like to be able to add leucine as a surface modifier or a different surface modifier to improve aerosolization. We need to understand the compatibility that we may have with the API. Not at all times can we use leucine, for example. So we're going to transition a little bit here now from a formulation approach to really what's the particle engineering aspects of designing an inhaled dry powder. So we've defined a formulation, we've looked at a couple of different formulations we want to make screen, mixing in different API loadings or bulking matrix or surface modifiers. Now, how do we actually make these particles? And that's where the particle engineering aspects come through and utilizing the spray dryer. So in each case, too, I just want to note that the particle engineering steps of the spray dryers are translatable from some very small scales to quantities, and we'll go through that a little bit in a later case study. But your process is really going to define the particle size and in combinations, the particle morphology and overall dispersion. So ideally, really what you want to have is a high throughput, high collection yield, while also balancing the different extremes within the spray dryer and the temperature profiles. So the slide that's presenting right now, it's really just a background about spray drying to kind of orient us all again. So there's a given physical situation. So as we fed our solution, the atomizer, you can see that kind of picture there in the top left corner. You're getting dispersion of individual droplets out of the atomizer. As those droplets now move into the spray dryer, you have evaporative cooling. So you have the hot drying gases coming over the outer surfaces. It's then evaporating off whatever solvent you had, whether that is an alcohol, a water, or a different organic material. And you have a skinning of the surfaces of the particle. And what you result in now is a nice dried spray dried particle. You will have some residuals, of course, because you don't get always effective drying through the process, but that will impact your yield. So in each case, really, though, you're looking at a mild temperature exposure. You do have higher inlet temperatures, shall we say, that can range anywhere from 120 to 170 degrees Celsius. But your outlet is really what defines what kind of temperature profile the active or the formulation is going to see. And typically, those outlets will vary a little bit about overall with a given mobility of the molecule. So you have short residence times, you have fast drying. In this case, you are rapidly solidifying the material from a liquid solution, and you can tune the material. So depending upon what kind of particle size you need, you can vary your flow rate, your atomization to give you the right particle in the end. And as I said before, this is all has precedence and you can translate everything from early feasibilities and scale it up to larger spray dryers. Some further information about spray drying itself and what we really care about as we go through the process. So you have multiple situations here. You have a spray solution, and again, as we said before, you're understanding the solubility of your molecule, of your excipients, what kind of stability does it have in those blends. We want to make sure we have the highest possible. You have pH concerns sometimes, different concentrations, and different solution-based interactions. That spray solution itself can be either a true solution, a suspension, or an emulsion potentially. You could feed one single solution, or you can have multiple solutions that are fed in. It do either different lines or mix just prior to atomization. Now we have the atomization, as I said, that defines the particle size, and really it's going to be based on the pressure itself that's being pushed through by the drying gas. The drying conditions will give you a particle morphology. So let's say you have something that will be very fast drying, so loose interaction with your solvent, you're going to have more of a spherical particle. There is a lot of information around how to define this, and there's a lot more literature out there for that. Something that's a little bit colder or slower drying will give you more of a wrinkled morphology. So this is really, again, based on how things evaporate off the particle. And your collection efficiency is really going to be based upon how can you as quickly define different densities in there. You want to make sure the gas will move out through while your solidified material will drop out into the collection vessels. And as I said before, you really want to try and increase throughput as much as possible. That can be tuned by flow rate or atomization or balancing different feed rates into the process. In each case, you want to make sure you can collect the right size. Giving surface area is important as well, right? So the more wrinkled the material may be, the more surface area this is, which will allow you for better aerosolization sometimes, but also then can lead to different electrostatic issues. Porosity is key. You can tune the spray dryer, with different organic solvents or different excipients to change the overall porosity or morphology of particles. And just to kind of tie this back a little bit too, the addition of leucine as a surface modifier, what it does really is it slows the water diffusion rate and kind of leads to change in the particle morphology, giving you a rougher particle and better aerosolization. So when we're developing a process itself, there's a couple offline tools we like to use as well. Top left there is a nozzle characterization or known as a nozzle test stand. What that does is look at individual droplets coming out of the nozzle and how that can be tuned. We can look at it changing flow rate or changing to the actual nozzle size, the air-liquid interface there. Additional things you really want to care about is what is your thermal constraint? So as we're collecting powder, is it sensitive to higher temperatures, lower temperatures? Because that really then will define what your drying conditions are. You kind of want to be able to use the highest outlet possible while ensuring stability, and that's really just based on the ability to kind of get off the residuals and also allow you for a better throughput, really, because you can put more flow into the condition. And water absorption, test this, or you want to test this to understand what the overall hydroscopic nature of your materials are. Again, you're trying to use excipients that will be less hydroscopic. That way your powder itself has better performance across different relative humidities. But you do this test to really understand, where is your product going to line up when handling the different RHs. We have done a couple of different cyclone CFD models to really understand separation of our particles, and again, based on density and gas flows. And in the end, really, you have a desired product profile that has the right particle size, has the right purity water content, and we're getting high product recovery. So you can use the spray drying process to meet different particle needs. I touched on before, too, a lot of the times this is going to be based on the API itself and the included excipients, but you can tune the dryer to meet each one of these. Of course, the easiest approach is some amorphous API, no excipients, is something that's solubilized in a preferred solute, and then processed through the spray dryer looking for effective dryer. Additionally, now you could add excipients. We talked before, either it's bulking or surface modifying. The surface modifying is usually critical for particle performance. You can do a top-down nanocrystalline approach. So you either wet mill or jet mill material, suspend it now in a nanosolvent with solubilized excipients, process that through the atomizer. The atomizer will add a little bit of shear and mixture, and it'll give you a homogeneous particle that will contain those individual crystals with the kind of amorphous excipient matrix. Now, you can also use the spray dryer to do a bottom-up approach. So you could have a solution that contains solubilized API and excipients, and then either use heat, which we'll go through the next slide, or additionally what you can do is have an antisolvent just mix prior to spray drying, so an inline mix procedure. So what you're trying to set up is a supersaturation event when spray drying. So those individual API materials will crystallize within a defined droplet with those excipients, and then have a particle that's going to contain, either a nanocrystalline or micron size API. And really, what defines the size of the API will be the supersaturation event, the active, and also the time kinetics of your processing. And lastly, you can have, of course, just a mixture of amorphous or crystalline products. In each case, we can have that defined within the spray dryer. We want to be able to make sure we ensure density and porosity throughout processing conditions in each case. So as I stated before a little bit, so there are ways to vary process to engineer particles. It's not simply always processing a material that's true solution and you're collecting amorphous material with excipients. What you can do is look at inline mixing of two streams, using a warm or hot process to help solubility and corresponding throughput or supersaturation. And additionally, what you can do is potentially use multiple nozzles or multiple solution feeds to help change your particle morphologies or particle throughput. So again, the inline mixing really can help solve a lot of solution stability concerns. You can put your API in its preferred, let's say, it's a small molecule organic to ensure stability. You have your excipients in a different stream. You feed those two streams just prior to spray drying atomization. So now you've limited that time course, and you go ahead and collect materials. And depending on the API performs, it may crystallize, it may stay amorphous. So that figure there is in the bottom left section of the slide. Another way is to use a warm or hot process. Now, it's similar a little bit, but what you're doing is enhancing your initial solubility and then also your throughput. So you have a solution. It has some solubility in the organic, and maybe your excipients are soluble in that. You feed it to the spray dryer. You use kind of a heater inline. That heater inline, of course, solubilizes the material, and then we can actually spray dry it. In each case, what we can do is perform or collect amorphous or crystalline product, and allows for really a broader solvent selection and can remove the need for jet milling. So on the bottom right side there is a diagram which kind of goes through the procedure of using a hot or warm process. So briefly, I also just wanted to give you an idea here of what those inline mixing or hot process may result in. So this is the bottom up. We're looking to supersaturate and recrystallize materials within the droplet. We're utilizing the concentration of solubility ratio. And so what we can do here in inline mixing, we change the time course a little bit to make sure we have large crystals that were formed just prior to spray drying, and then we got coated with the material here during the process. And this can impact your overall kind of ordering or of the crystal state. You kind of see that in the bottom left corner of that graph, what the API melting temperature is, what a physical mixture looks like, the inline, which had a slightly lower melting temperature, and then the hot process. The hot process formed nanodomains. We can see in the top, or the kind of image on the right-hand side here, that particle is made of a bunch of nanodomain API products. And really, in each case, we want to understand, by using the TXRD, what is our energy state of those different crystalline forms. So with all these kind of tricks and things you think about during process, it doesn't really mean anything unless you're able to scale it up and keep it consistent. And so a lot of the times we'll translate things that are done on a lab scale strier, which looks at 300 milligrams to 100 grams for formulation screening, moving to now a PSD1, which is just a larger spray dryer, which allows for a higher throughput, and that's why you have higher batch sizes in this case. And then even translating that to clinical or commercial manufacturing, where you need kilograms of material. You can use the PSD1, or we do have a PSD2 in each case. So scale-up can be relatively straightforward. We do have models and just a function of mass transfers, so you're balancing out what is your feed rates and your temperature profiles here in this case. And again, as I said before, by adding multi-head nozzles, you can increase kind of collection efficiencies and powder masses. So now that we have a formulation defined, selected, we find the right process conditions, there's a couple different things actually completing the product here. And you know, what's really important a lot of times is, are you going to go a route of blister packing or encapsulation in this case? And so a lot of times we'll go to encapsulation. It's easier a little bit on the early feasibility stage. And you care about what those capsules will do for you. So you want to make sure that there's a lot of or no interaction really between your powder and your capsules. That can be a function of moisture and the lubes that are used for different capsules. You want to have the highest emitted fraction at all possible. And a lot of the times what you do is you fill these into kind of size 3 capsules are very well used, and again, it's more of a function of what's available. And the idea here in this case is to use the simplest device to help you deliver your product. And in each case, what we hope to do is develop a robust formulation, a robust process, so that the device itself doesn't make as much of an impact in how the powder performs. And so the last part of this section here, again, is just defining the total package, just taking understanding of your formulation, what is your process, what is your capsule or your blister, what is your device. In each case, as you move through these components, you need to think about these all together. They are really what's going to be your final product. And so you want to ensure that you have a successful product by having a stable, robust, and simple process that the patient will ultimately be able to use. So the last section of today's webinar, what we'll go through is 2 different case studies that will help you a little bit understand what we would think about when we go to formulate or design a process for different products. So the first case study now is looking at 2 different molecules that are actually topoisomerase disruptors. Our real main goal here was to define a scalable process that will help aerosolization of the dry powder, and we're trying to target local cells themselves. And really want no systemic exposure of the material. Both molecules were highly potent and required low dose because of that. So, as I said, the molecules themselves have the same mechanism of action, but their physical properties were different. So because the physical properties are different, we need to define a different formulation. So the first molecule, small molecule, higher TM, and then a lower TG. The log P in this case, lower than 2, is kind of a little bit of a lower level, and that helps us a little bit, but it is water insoluble. Molecule number 2 now has a lower TM than the first one, a higher TG, and a higher water solubility. So based on these kind of physical properties, our general approach here was, okay, well, we want crystalline API for the first molecule because we don't believe it's going to be stable in the amorphous form, even though it is a low active loading. And we'll use those excipients to provide kind of the amorphous matrix, particle properties, things like that. Molecule number 2 now, because it has such high water solubility, it's relatively stable in that, has a high TG, really lends itself to more of a amorphous material that can be dissolved with functional excipients in that aqueous media and have that kind of final total package that is just amorphous. We did use a little bit of the buffer salts to help decrease the pH, the preferred range for the molecule. So the next slide here just gives you a quick overview of that crystalline formulation approach. Again, as I said, we were looking at developing a crystalline API. So we used this bend log dryer, in our case what we term it. It's just a small-scale spray dryer that was tunable to give us the particle performance we needed. We went ahead and nanomilled the API by using a mixture of some poloxamer polysorbate and phospholipids, those surfactants, and made really a suspension of those nanocrystalline materials. In this case, we had dextran as a water-soluble high TG amorphous matrix. So that was fed into a single solution, then into the dryer, mix it with the drying gas, the atomizer through the drying chamber, had our cyclone separator to collect our particles. The bottom right side there, you can see some representative SCM images. And again, the idea was to have crystalline API inside the particle, having some of those surfactants on the outer portion of the particle with a bulky matrix dextran in this case. Some of the analytical from that approach here in this case, we did do some SEM and TEM to understand that we did contain nanocrystalline API embedded in those micron particles. So you can see those images on the right-hand side of this slide. Did some aerosol evaluation to ensure that those surfactants and the bulky matrix were able to define our particle performance. This was loaded into a size 3 HPMC capsule, actuated at just a monodos device, and then looked at for properties. So we had a nice mass median aerodynamic diameter, MAD at 2.65, a low standard deviation, fine particle fractions almost 70%, and emitted fractions of 90%, which is a good profile for a product to be evaluated. And the material was then sent out for in vivo studies, which did confirm the API released and got targeted exposure on the outer portions of the lung epithelial. Molecule 2, really in this case, we did an amorphous API formulation. Again, high water solubility. So all of those materials were mixed. The API had trehalose and leucine in water. So it was a true solution. It's fed to the spray dryer. The idea in this case was to have leucine recrystallize upon spray drying, keep trehalose amorphous, and the drug itself amorphous. Process recovery was 92%. Water content was defined really by the residual amorphous trehalose. And then the particle size itself hit our target product profile. And you can kind of see that here in the specifications that were laid out in the slide. The representative image there is on the bottom right side of the slide as well. So there was some corresponding analytical of this formulation approach. We confirmed that there was the right chemical potency and purity of the molecule. Again, did have the amorphous API and trehalose. The X-ray powder diffraction pattern there is shown on the right-hand side of the slide, where you can see then blue corresponding to leucine, the red, the API, and then the sample only containing diffraction from the leucine material. Had a nice high TG, again defined by trehalose added as the excipient in this case, so right around 118 degrees Celsius. And the aerosol performance was consistent with the other formulation we just discussed. MAD around 3, emitted fractions 94%, fine particle fractions of 62%. In each case, we were able to successfully manufacture those formulations and were sent out for in vivo comparisons. So what I'll briefly do in the next couple minutes is also show the second case study. And this case study is more around preserving scalability and properties across a different API load. So the goal was really to understand how we can tailor processing conditions to ensure that we get the same powder performance, and then how can we also alter the formulation and then corresponding in the process to still give us the same amount of or same powder properties. What this does is allows you to do a lot of dose range flexibility. It can be based on either how much powder you need or what is your formulation, or also in this case we did look at fill mass when aerosolizing. So model compound was albuterol sulfate, formulation excipients, we had a trehalose, leucine, and sodium chloride for reasons which I've discussed previously. Active loadings were either 10 or 40%. And the spray drying equipment, we performed this either on a lab scale spray dryer or on the PSD1, so pharmaceutical spray dryer. So this slide really is representing a couple different batches we did here. So on the lab scale spray dryer and then comparing that to the PSD1, we varied batch size on the lab scale spray dryer. In each case, what we were able to do is we tailored the process to give us the same exact properties across those different scales. Process yields just increased a little bit with batch size and scale, which is great. Water content, though, is consistent. MAD was the same. GST, emitted fraction, and fine particle fraction, all within range. And you can kind of see those results from the NGI at the bottom section of this slide. Again, what this does is allows you to do different, or to feel comfortable when moving from small-scale feasibility batches to larger pilot-scale materials. We went ahead and filled this material with an Exceladose and compared that to hand fill as well. A couple different fill volumes. The idea here is to ensure that the powder will perform across different conditions. So we had hand-filled material, 5 mg, 10 mg, or 20 mg filled with the Exceladose, and we're able to show that the powder performance was consistent. MADs were close. GSD, emitted fractions. There was some variation in the emitted fraction, but really all the times that a function of there's powder coating within a capsule. So a 5 mg fill is usually pretty low for a size 3 capsule, and there was some powder compaction there, and that's why it decreased slightly. But overall, the profile in each case was consistent. So again, you can take material, you could vary the fill mass to get your dose titration and understand what your product profile is going to look like in the end. So another wrinkle to this is what we did is look at varying active loading. So we changed it from 10 to 40%, altered the process slightly, and in the end, across both scales, we're able to show that the powder itself performed the same. And again, it's a function a little bit of the formulation we selected and some tweaks to the atomization or flow rate, and also now when you go to the larger scale, the mass transfer, and allowing you to collect material. And so you can, again, dose materials that have lower active or higher active or different fill mass or different scales and still give you the same properties. And I'm just showing this in the bottom part of this slide, the overall aerosol distributions. There's some corresponding analytical just to confirm those results. Again, this is from a Malvern, so laser diffraction, looking at particle diameter. It was consistent across both formulations and both scales. And you can see those particle morphologies of each batch as well were consistent across each one of those batches. And just to kind of let you know as well, as we moved to the 10 to 40% active loading, all we did was vary the bulky matrix trehalose while keeping the leucine the same loading. So with summary here in this case, we've gone through a lot of things through this webinar, but spray drying itself really is an enabling technology. You can design particles that can be delivered to the lung. There'd be a variety of API classes. We could do small molecules. We can do biologics. In each case, you're just tailoring what those excipients will need to be to stabilize the material. and altering process conditions to, again, ensure that you're not having any degradation of any sort. Formulation considerations are always based on the active itself and the physiochemical properties and also your PK/PD response. Processes can be altered to deliver the desired particle size. We can use single solvents, co-solvents, inline mixing, a warm-hot process in each case to help utilize increased throughput or to get you the right particle morphology necessary for aerosolization. In all cases, these approaches are really robust. They're scalable, can be sparing. As shown kind of that last case study, you do 300 milligrams and still get you material that's going to be representative when you go to larger batches of PSD1. And kind of through this development, we've been able to really get the right knowledge and equipment and design in place to be able to optimize formulations, process, manufacturing, be able to encapsulate, provide the given analytical evaluations, and also then corresponding to your GMP release. And just kind of the end note here in this case, and while you're designing your formulation and process, you really always want to think about how appropriate is the phase of your program. So is it feasibility? Is it moving toward now clinical? Is it even going further from clinical and commercial manufacturing? And you really want to ensure efficiency and flexibility throughout the process to not be surprised in the end. So with that said, I just want to thank you again for listening to the presentation. I hope it was informative and educational. Of course, there is a lot more detail that's out there. And in any case you would like to contact us, please do. We'll open it up here for a couple minutes for questions. Thank you again.
Thank you so much, Mike. I think it was a very great webinar. Unfortunately, we do not have a lot of time for questions. So my suggestion will be, well, I've compiled already a number of questions, some of them related to the formulation, some of them more related to the process, stability, or scale-up challenges. I don't think we will have the time to go through all of them, but be sure that all those questions will be submitted to Mike, and we will document some answers and make sure that we are following up accordingly with every one of you. Again, thank you very much for attending the webinar. Do not hesitate to contact either Mike or myself. The email address is fairly simple, just first name dot surname @capsugel.com. And we hope to see you soon in the next Capsugel webinar. Thank you.